advertisement. So I want to present today the recent work that we did at the CCEM about how we can establish a correlative workflow to examine human bone tissue across multiple length scales. I also want to talk specifically about the proper sample preparation of biological tissues for electron microscopy, because I think especially for the past year or so, given the expansion and the new, the new instruments and equipment coming into the CCEM, especially for the cryo-related equipment, I think there is a huge potential down the road and in the future, particularly for proper sample preparation for biological tissues and how we can utilize those new equipment for electron microscopy imaging. So to start with, the overall motivation for my research in general, if I have to use one word to summarize it, it will be the hierarchy. So I want to actually pull out a laser pointer. There we go. So the structure hierarchy, pretty much we can observe everywhere in our daily life, ranging from this kind of a tall architecture, which is actually built upon wood. A natural material in itself also has a very complex hierarchical structure. The word hierarchy actually originated from this Greek word, hierarchy. So I will not pretend I actually read Greek, but essentially it refers to any system that is comprised of entities, either physical entities or abstract entities, and those entities would rank either one above or below each other. And when it comes to the living system or biological systems in nature, we know that the structural hierarchy actually plays a very essential role in terms of biological and mechanical functions. So here is an example of the gecko feet. A few decades ago, there was a really intense research interest in understanding how the gecko feet can attach to the smooth glass surface so strongly, but the gecko can also easily detach the feet from the glass surface when they want to. So the researchers find out what happens is actually comes down to the complex structural hierarchy on the padding of the gecko feet. So if you zoom in, you will see the tip of the gecko feet is comprised of this numerous hair-like structure. So those are called the setae. Each one of those setae is actually further branched out into even smaller structure, and those are called the spatulae. The spatulae, eventually, they can interact with the surface at the molecular level. So eventually, it's a van der Waals force that really directly bond the gecko feet and the glass surface. And that is the mechanism that creates such strong bonding between the gecko feet and the glass surface. So this is just one, very typical example in terms of how structural hierarchy actually plays such an essential role in terms of the biological and mechanical function. And we know that in nature, there's so many examples ranging from the pangolin scales that have those multiple layers to the porcupine spines, to turtle shells, to rhino horns. So all those nature-derived materials have a very complex hierarchical structure. So for me, in terms of my research, I'm particularly interested in understanding the hierarchical structure of our own skeleton. So we know that our own skeleton have multiple functions, ranging from protecting our internal organs. It can also store critical nutrients and minerals and proteins. It can also, allow us to, have multiple movements, especially in our joints. So all those functions, particularly the mechanical functions of our own skeleton, comes down to it's a very complex hierarchical structure. So for the people who are not familiar with the hierarchical structure of bone, so I will just use the human femur as a typical example, just to demonstrate how complex the structure is at different length scales. So here, as you can see, this is a longitudinal section of a human femur. On the outer layer, you have this very dense, cortical bone layer that surrounds the internal spongy trabecular bone tissue. If we further zoom in into the cortical bone region or into the trabecular bone region, you'll see at the microscopic level, it's comprised of this numerous cylindrical shaped units, and those are called the ostium. And of course, embedded within the bone tissue, you see those dark, like pore structure. Those are the cavities that host the bone cells. Within each one of those ostium is actually comprised of this highly organized, parallelized layers of mineralized collagen fibrils. Of course, this is a simplified version where you see those mineralized collagen fibrils, they will align with each other within each layer, and they will rotate between the different layers within this each one of this ostium. And at the very fundamental level, this is at the nanometer length scale, bone is comprised of the collagen molecule and nanometer-sized mineral particles. So that's how bone structured from all the way from the macroscopic level to mesoscale to microscopic level and all the way down to the nanometer length scale. And this is already very simplified version. In reality, the bone tissue is much more complex than what is showing here. And the latest study has demonstrated that there are actually up to 12 different levels of structural hierarchy when it comes to describe the bone architecture and structure. So for us to really understand just to provide a comprehensive picture of how bone is structured across the different length scales is non-trivial. There are a couple of major challenges. The first challenge is actually how we can properly prepare bone tissues and also other biological tissues, in particular for electron microscopy imaging. So in contrast to what you might assume that typically, you can just put a piece of biological tissue like it was showing here, a bug on top of the alumna stub, it actually involves more complex steps. In particular, if we want to really preserve the tissue structure relatively well and close to its native state. The second major challenge, of course, as we all know, and this is not just pertaining to biological tissue, but in other materials in general, is this competition or compromise between the resolution and the imaging volume. We know that if we want to resolve better structural details with finer resolution, we typically won't be able to image with large tissue volume. And on the other hand of this spectrum, if we want to encompass large enough tissue volume, we won't be able to resolve as many structural details as possible. So one way to really overcome this challenge is to combine and integrate different imaging modalities through a correlative approach so that it can provide a more comprehensive view of the material structure across all the different length scales. So the overarching goal of this particular project is that we want to establish a correlative workflow to examine human bone structure across multiple length scales. So for the talk today, I will divide them into two parts. For the first part, I want to specifically talk about the proper sample preparation of biological materials for electron microscopy imaging. So I will touch upon the chemical fixation, which is kind of most accessible to most of the labs because you don't need to use a very fancy equipment. But I want to focus more on the second, sort of the cryopreservation and the cryofixation part, because I think this is where the CCEM has, at least for the past year or so, have acquired those cryo-relevant equipment that can perform such a task. And for the second part of my talk, I will talk about the recent work that we did at the CCEM, where after the sample preparation, how we can examine the human bone tissue across different length scales using a correlative approach. So first, I will talk about the sample preparation of biological tissues. As I mentioned briefly, we typically start with a chemical fixation for biological tissues because this is the most widely available for most of the camps, like wet labs, and you don't need a really fancy equipment for this kind of chemical fixation and preparation of biological tissues. So briefly speaking, we typically start with some kind of fixation of the biological tissues. The idea is to use those fixative to stabilize the tissue structure. So for example, here, we typically use glutaraldehyde and paraformaldehyde to cross-link the proteins and specifically the collagen in the bone tissue to stabilize the structure in general. And sometimes we might also use osmium tetroxide to, it's not only stabilize the cell membrane, the carbon-carbon double bonds in the cell membrane, but it can also stain the cell membrane to increase the contrast under electron microscope. The general consideration for the fixative when you prepare the solutions, of course, is the pH that should be more or less neutral, and also the molar concentration as well ionic concentration to make sure that or at least to minimize the osmotic shock. And after fixation, we typically perform dehydration because for biological tissues, for example, in the cells, more than 80% of the cells are actually water. So we have to replace the water very gradually with some kind of solvent. In this case, it will typically be either ethanol or acetone. And after dehydration, we will infiltrate the biological tissues with a solution that's typically a mix of polymer. And this polymer, the idea of this infiltration is to use this liquid polymer to replace the liquid phase within the tissue. And when we choose a proper polymer, the consideration is the viscosity of that polymer and the size and the density of the material, especially the cavities inside of the biological tissue that need to be infiltrated by the polymer resin. And of course, after the infiltration, we'll harden those polymer either on the heat or under UV light. So in general, we could either pick epoxy as the polymer or acrylic as the polymer. In this case, we typically go for epoxy or PMMA because they are typically more stable under electron microscope. So if we compare the tissue that's typically prepared under chemical fixation, we'll realize there are a lot of limitations, or I should say artifacts. The reason being, regardless how gentle we are when we fix the tissue, when we dehydrate the tissue, and when we embed the tissue, there can be always shrinkage, or in terms of the cell proliferation, and we might even observe some solvent extraction or dissolution of the unstable precursors after the chemical preservation. So here is a very classic comparison between a chemically fixed yeast cell compared to the right hand. This is a cryo-preserved yeast cell. So I hope you can appreciate how different in terms of the cellular architecture. So here is the cell membrane preserved under the cryo condition, and you can observe all the details and compare to this one, which is chemically fixed. So that's why the next part of my talk, I want to talk about the cryopreservation and cryofixation. The reason we often talk about cryogenic preparation of the biological tissues is because of the problem when we freeze water. So I think we all have the experience when we buy frozen fruits, for example, frozen strawberries from the grocery store. we'll realize that typically this is how the fruits look like when they are frozen. And once we defrost the fruits and back to room temperature, they would look like this. So they still have the water content, but they just don't look as fresh as before they're frozen. The reason is that during the freezing process of the fruits, it's typically relatively slow process, even though we call it as a flash frozen, but still it's relatively slow. So during this process, the ice crystal, they will form, they will actually damage the cell structures. And when those ice melts back into water, the cell membranes and the cell walls have already been compromised, and the intracellular contents will be released. So they were never as intact, the cellular architecture was never as being intact as before. So really, to preserve the cellular architecture as native as possible, we have to perform cryogenic preparation. So this diagram actually demonstrates the impacts of different freezing speed. So when the freezing speed is relatively slow, the ice crystal might form either outside the cells, and that will cause the dehydration and cause this osmotic imbalance of the cell membranes and disrupt the cell membranes. The ice crystals can also form inside of the cells. So those are called the intracellular ice formation. They will damage the cell organelles inside of the cells. Only if we increase the freezing speed up to a point, the ice crystals will not form. So we'll only form amorphous ice. So this process is called vitrification. With vitrification, we can either use a really high speed in terms of freezing. We can also add a cryoprotectant agent. So the cryoprotectant agent can actually lower the freezing point of the water. So that can be also very advantageous when it comes to cryogenic preservation of the cells. And of course, the same ice crystallization can appear during the warming process, if the warming speed is really slow. But today I will be only focusing on the cryogenic preparation side. So when it comes down to the cryopreservation of the biological cells or tissues, in general speaking, we can either resort to plunge freezing or high pressure freezing. So at the CCEM, we actually have access to plunge freezing. So this is suitable, for example, if you have cells that you want to preserve under cryogenic condition. The reason being the plunge freezing has a limitation in terms of the thickness of the sample you can prepare. Typically, the cells are in the magnitude of 10 micrometer, and that's suitable for plunge freezing. If you have a thicker sample, like hundreds of micrometers, then you have to resort to high pressure freezing. So here's a few pictures as an example of a typical plunge freezing machine from the FEI. So I think CCEM has access to a similar plunge freezer. It's the VitroBot. So essentially the samples or the cells with the liquid will deposit on the grids, and this grid is holding between the tips of this tweezer, and then you will plunge freeze the sample into either liquid nitrogen or liquid ethane to the liquid nitrogen temperature with the cryopreservation. But if you have, let's say, thicker tissues, about 200 to 300 micrometer, that won't be suitable for plunge freezing. In this case, if you want to freeze thicker tissues, you have to add higher pressure, because higher pressure can also lower the freezing point of the water. So as you can see in this diagram, when we increase the pressure up to a point, this is about 2000 bar, we can significantly lower the freezing point of the ice. So typically for high pressure freezing, as demonstrated here, what you do is you will cut out a thin slice of tissue typically thinner than 300 micrometer, and sandwich the tissue between the two planchettes. And this is how the planchette looks like. And then you put the planchette into this carrier of the plastic carrier, and then you will need to push the entire carrier into this high pressure freezing machine. So the machine will instantly freeze your sample at very high speed, but also add the 2000 bar pressure, and the sample will be collected into the liquid nitrogen door on the side and where you can collect it. So the major challenge, if you high pressure freeze your samples or plunge freeze your samples, biological samples, and directly take those cryogenic condition prepared samples to electron microscopy imaging, what you will see is something like this. So this is a cell that's imaged under EM and cryopreserved. And you will notice the contrast is very low. That's because the major content is water. And the cells is also made up of those light elements because it's all organic. So it's mostly carbon, nitrogen, and oxygen. So if we want to increase the contrast of the cellular tissue, we have to use some heavy metal staining to really help to bring out the contrast. But of course, the sample is already cryogenically preserved. So how can we stain the sample with those heavy metals? So the next step is actually to use a freeze substitution with some heavy metals, for example, the osmium tetroxide or uranium acetate. So I think at the CCEM right now, we don't have access to the freeze substitution. So I will just very briefly talk about the purpose of the freeze substitution. So here is a typical example with the freeze substitution. Essentially, once the sample is prepared under cryogenic condition, either on the grids or sandwiched between the planchette, they'll be stored with the liquid nitrogen. Then you have to transport your sample into a vial that contains the fixative and those heavy metal staining agents, as well as dehydration agent, mixed together with the liquid nitrogen, because the sample still has to be under the cryogenic condition. Then you will put this vial together with the sample and with all those cocktails mixed together into the chamber, into a freeze substitution machine, as an example here. This machine typically will pre-cooled to a certain temperature. In this case, it's minus 120, and will gradually warm up to a certain degree. This is a minus 90 degree, so that the cocktail will start to melt. And for example, here, the acetone and other heavy metals, they will start to enter the cells and tissues to replace the water. And typically, the whole process will go through several steps, very gradually warming up all the way to room temperature eventually. And the idea is to let those fixatives and heavy metal agents to gradually enter the cell's tissues during this warming process. And at the end of this diagram, the samples, so here's a picture of the high pressure freezing samples and also went through this freeze substitution process. This little dark piece here, that's a tissue sample stained with the osmium tetroxide. At the end of this whole process, the sample will already be embedded within the resin. And then you can essentially trim down the resin and expose the sample and then perform the electron microscopy imaging just at room temperature. So that is about the freeze substitution. And of course, the other way after, cryo preparation of the sample is to directly take those samples after vitrification to cryo imaging. So in this case, we basically have to find a proper way and workflow to directly transport those vitrified samples from either the plunge freezing or the high pressure freezing machine to the cryo imaging, so cryo TEM or cryo FIB-SEM. So the way to do the proper transportation is that to resort to some of the equipment shown here, which the CCEM has actually acquired since last year. So that involved the Leica VCM, VCT, as well as the Leica ACE. So I will talk about briefly how this transportation happens. So briefly speaking, once the samples is in the liquid nitrogen vial, we need to find ways to transport those samples into this VCM machine and then utilize the VCT arm to bring out the sample, either the grids or the planchette into the VCT and bring the VCT to the ACE machine. So ACE is for the coating of the sample surface. You can also fracture open the planchette to expose what's underneath the sample, and then use the VCT again and then transport the samples that's after coating to either cryo-TEM or cryo-SEM. So I have a short video just to demonstrate very quickly how the transport of the sample happens using the VCM and VCT. So here is a top view of the VCM machine where you can see this is the chamber of the VCM, and those are some of the cryo-preserved samples in the planchette. And so, of course, the chamber is filled up with all the liquid nitrogen. So you have to bring all those planchette together with the samples still inside to the stage here. And then you have to tighten the stage, or basically to tighten the grids so that they will be fixed on this stage. And then you have to find ways to rotate this stage so that they will be facing upwards, where you can see an opening there. So this opening part is where the VCT arm will come in later. So what's really critical here is that all the tools you see here, they have to be pre-cooled to the liquid nitrogen temperature. So all the tweezers and everything, you see all those slots here, you basically have to put them in and pre-cool them for at least a few minutes so they can reach the temperature equilibrium. So once the stage is turned to the side that's facing this opening here, you will see, so that's the rods coming in from the VCT. I think there will be an enlarged view on the side as well, so you can see it. So yeah, that's the rod coming in, and we'll push it in to grab the stage. So what's really important here is the direction where you turn the rods. Apparently, when you turn the rods in certain ways, you might lose the stage and release the stage, and then you turn the other way, and that will grip the stage. So once the stage with sample is in the VCT, then you can detach the VCT and attach the VCT to the next equipment. So here, I'm not going to demonstrate what's happening next, but typically you can either attach the VCT to the ACE machine to perform coating, and then bring the VCT arm again to your cryo-TEM or cryo-SEM. So that's all about the sample preparation. And for the next part of my talk, I want to talk about after the sample preparation, how we can utilize a correlative workflow to examine human tropical bone tissue utilizing the different imaging modalities here at the CCEM. So here, the examples will be on a human bone tissue that's prepared at the room temperature using chemical fixation method. So to give you a bit context about why we want to perform correlative tomography, in the past, there has always been an interest in, utilizing a correlative imaging approach to really examine tissue structure and other traditional material structure at different length scales. But it was mostly done at the 2D level instead of the 3D in terms of like a tomography. So we all know that 2D imaging always has these limitations in terms of really give us a full picture of the tissue structure in 3D. And in the past few years, there's some studies have been done in terms of 3D correlative tomography, but it was mostly geared towards engineered materials or semiconductors or even implants. And there was I wouldn't say there's any studies have been done so far target towards the biological tissues. So the major challenge in terms of the 3D correlative tomography is to first establish a proper image registration process across different imaging modalities that can help us to perform some site-specific preparation of the sample. So in terms of the correlative imaging workflow that we have established here at the CCEM is that we first utilize the crossbeam laser fib to create a customized grid on top of the sample surface. So here is an illustration of a piece of human trabecular bone tissue that's embedded inside of this plastic resin. And then we use the laser fib to create this grid on the sample surface. We then, put this entire sample into the Versa 630 XRM to acquire an overview of the entire sample. We can further zoom into specific region of interest using, again, the XRM and acquire a high resolution but more targeted scan of that specific region of interest. Then we took the sample and put it back into the crossbeam system again, and we can perform site-specific preparation based on the scan acquired from the XRM and utilize the correlative workspace inside the crossbeam system to specifically target this selected region of interest and expose the cross-section. And finally, use the plasma fib SEM to examine the structure at the nanometer length scales with the 3D tomography. So more specifically, the first step where we create this very customized grid system on the sample surface, we use this crossbeam laser fib. So as you can see in this image panel A here, that's a piece of plastic embedded, human bone tissue inside. So you can see very vaguely, those are the human bone tissue inside of the resin. And we actually combined some letters and numbers on the two sides of the grids on the sample surface so that we can specifically, identify some region of interest within the grids. So what's really critical when we design these grids on the surface of the sample is that these grids need to be able to be visualized not only under electron microscope, but also under XRM, so that we can really correlate between the two different imaging modalities. We then put the entire sample into the XRM where we can scan the entire sample, including the grids together with the bone sample. We can also, start to target specific region of interest that we want to examine further at the smaller length scale. So in this case, it's this, blue shaded region that's a single bone trabecula. So we can directly locate this bone trabecula in terms of 3D spatial location with respect to the sample surface, but also in terms of its spatial location, with the facilitation of the grids that we created with the cross-beam system. And with the high resolution imaging within the XRM, we can, appreciate even finer structural details within that selected region of interest, where you can see those small pores, dark pores. So those are the cavities that host the bone cells. You can also see the different, gray levels. Those gray levels are essentially a reflection of different mineral content within the bone tissue. And this step, which is very critical following the XRM scan, is that we can import the XRM data into the crossbeam system, and we can overlay the XRM data with the XEM image of the sample surface acquired in the crossbeam system, and utilize the Atlas correlative workspace. We can directly register those imaging data together. So what's really critical about this step is that because we can overlay the two different imaging data set, and particularly the XRM data is performed in 3D, we can really decide where to start the milling process and what is the field of view in terms of laser fab preparation. So as you can see in the image D here, this is a cross-section of the bone tissue that's already being exposed after the laser fab. And as you can see, the cross-section of this bone piece is very irregular, and the width of this bone piece is wider than what you can observe on the sample surface. The central line of this bone piece also shifts compared to what you can observe from the sample surface. So when we overlay the XRM data in 3D and the SEM image using the correlative workspace, we will be able to decide, okay, the sample tissue will be much wider than what we can see here. And this helps us to decide exactly what is the field of view we have to expose using the laser fab and where we want to laser fab and expose this cross-section. So that's what this correlative workspace coming from. And after we use the laser fab to really quickly remove the excessive material and expose the cross-section, we can also fine polish the sample cross-section. Then we basically use the plasma fab SEM and further examine the structural details and cellular architectures at the even smaller length scales, where we can see not only the cell cavities as shown here, but we can see those nano channels. So those are the channels that connect the cells. We can also observe those mirror clusters. So those are the very ultra structural features of the bone tissue in this case. And finally, we can also correlate the different imaging modalities, in this case, in between the XRM and the plasma fab SEM, where we can see the similarity between the two different imaging modalities based on the unique shape and location of those cells. But of course, the PFAB SEM can provide much finer details and sharper images compared to the XRM, but the limitation is, of course, the tissue volume, where the PFAB SEM will have less imaging volume compared to what the XRM can provide. So this is what we have recently established at the CCEM, where we can use this correlative workflow to examine the human trabecular bone tissue. And what we have also recently started is that we're hoping to utilize this correlative approach to examine even more complex biological systems and apply to some critical and clinically relevant topics, for example, the osteoarthritis or OA. So because we know that for osteoarthritis, typically it was regarded as a disease that involves the wear and tear of the cartilage in your knee joints, but it's actually much more complex because the knee joint involves multiple tissue structures at different length scales. It involves not only your cartilage, but also your calcific cartilage, and also the interface between cartilage and the bone tissue are buried underneath the cartilage. So the plan here, and what we have recently started at the CCEM, is to utilize some established animal model where we can observe the progression of the OA so that we can start at the overall view utilizing the XRM to scan the entire knee joints at the different stage of the OA. And then we can further zoom in to particularly target a specific region of interest that is more critical and involved during the progression of the OA, and further examine them at the microscopic level and also at the nanometer length scale, utilizing the techniques and the correlative approach that I just mentioned, and finally to really identify some of the critical structural changes that involved during the development of the OA. So to give a brief summary about my presentation today, for the first part of my talk, I talked specifically about the proper sample preparation of biological tissues for electron microscopic imaging, because that's like the starting point for any examination of the structural features of biological tissues. And particularly because of the new acquisition of the cryo-relevant equipment at the CCEM, I think there's a great potential for cryopreparation of biological tissues at the CCEM in the future. And for the second part of my talk, I talk about the recent work that we did at the CCEM in terms of how we established this correlative workflow to study the human trabecular bone tissue at different length scales. And we demonstrated that the laser fib can be a really powerful tool to customize this grid patterning to help facilitate the image registration between the different modalities. And it can also facilitate this site-specific sample preparation utilizing the ATLAS-5 correlative workspace. And finally, we hope that this kind of correlative approach can be utilized for even broader range of biological materials and even for more complex biological systems for more clinically relevant topics. And last but not least, I want to thank the funding agency. I also want to thank the Grantfield Research Lab and of course the CCEM, where we have carried out all this work. So just for a side note, I'm actually moving to the University of Virginia in about 1 month, but I will definitely keep all my collaborations here at McMaster, and I hope to continue some of the work that I just mentioned today. And I welcome any questions if there's any. Thank you.
Great. Thanks, Toni. That was a really nice presentation, and we're sad that you're leaving.
I know.
If anyone has any questions, please feel free to put it in the Q&A and the chat. I guess I have just a quick question to start it off. So you, in terms of resolution, the highest resolution you've gone to so far is on the PFib. Is there any desire to push that even into the TEMs or atom probes since the sample is already in the PFib? I'm just wondering what your thoughts on that are.
Yes, we actually do have a few. I mean, I have been talking to Travis, and I have several ideas about not only the PFib, but we have also talked about how we can correlative the volume, the 3D volume imaging, either in PFib or just the gallium fib SEM, and then do a TEM lift node within the same volume or adjacent volume and do a TEM tomography, so that we can really bridge all the, even, of course, the APT part is out of my expertise, but I think there's definitely some interest there, like how we can go basically all the way from the XRM all the way down to the atomic level. Yes.
Yeah, that would be, I mean, even getting to like the all the way from the X-ray to the fib is super cool, but then I guess that's even adding a new resolution and a new dimension that is really only available once you get down to the TEM.
Yeah.
How hard do you think it will be to like prep samples like that?
I think for APT is probably the most challenging. From volume fib SEM to TEM lift out, I think that's pretty, at least each of those approaches is pretty established. It's just like we haven't done any correlative work yet.
Yeah.
So I think that will be, I would assume that would be easier than going all the way down to APT, because the APT itself is already very challenging in terms of sample preparation. Yeah.